Today, I will be talking about my ongoing work on building a multi-state evolutionary model that can be used to quantify cellular plasticity during tumor evolution. So, cellular plasticity has a strong implication on prognosis of cancer as well as subsequent treatment, and therefore, it is an important issue to address. Cancer has been studied from an evolutionary perspective, which is something that I have been working on for many years now, based on the postulate that cancer originates from a single cell of origin. It is believed that, and it has also been shown that mutations accrued in stem cells over our human lifespan, causing certain clones to be under positive selection. They become clonally expanded under such positive selection because cells carrying these mutations are then biased towards more frequent self-renewal, and therefore they grow exponentially in the stem cell pool, thereby enhancing the chances of them further occurring further mutations, which can then lead to even higher proliferating rates. And eventually, this can cause cancer in some cases. However, even though this is this has been a very insightful approach in many ways, allowing us to quantify driver mutations, finding missing selections in cancer and precancerous tissues. This is less applicable during the later evolutions of tumors because during later evolution of tumors, there is a stronger selective advantage. There is a more dysregulated genotypic as well as phenotypic landscape for cells. And therefore, under such during such stages, it becomes less relevant, and it becomes more important to consider the phenotypic transitions more explicitly at these stages. In particular, in cancers like liver cancer, colorectal cancer, so liver cancer is something that I'm currently working with in the lab, in the small lab at Hong Kong University. And therefore, this is a background of why I am doing this. And because of what I've just told you, I am currently building a evolutionary model to model the later evolution of tumor with an explicit modeling component for the phenotypic transitions. And um, this is in combination with recent advances in the field. Previously, current methods that are used to understand the phenotypic transitions and phenotypic profiles in tumor evolution is mainly single time snapshot. So what we usually do is we look at the bulk or the single cell RNA sequencing profile of tumors and use different algorithms in trying to in order to understand what is the underlying dynamics that happens. For example, we have splice dynamics used as a method to infer where the cells are transitioning towards over the single cell RNA sequencing landscape. However, recently there is an exciting advancement. So there are technologies that allows us to probe the evolution of tumors using CRISPR-edited barcodes in mouse models. So what we have is that we inject, so that's not us, but using this technology, what one can do is you can use barcoded cells with CRISPR barcodes and inject them into mouse models. Once you induce the CRISPR edits, then random edits will occur. So this acts like natural mutations, marking the evolutionary trajectory of the clonal evolution itself, which means that at the end point of this experiment, when we harvest the tumors, we can then reconstruct the evolutionary history based on the genetic relationships of these cells. And what is exciting about this technology that I'm showing here, published several years ago, is that they not only just allow a readout of the CRISPR barcodes at a single cell level, but also they are able to provide a single cell RNA readout at the same time. So by just mapping the CRISPR barcodes from from cells that are collected with the single cell RNA sequencing result from 10x, it means that we have both the genetic CRISPR edited barcode information as well as the transcriptomic level information for every single cell. And using this data, I'm going to test the multi-state evolutionary model that I'm building. In this data, they have recovered more than 40,000 single cells from a single mouse across several different sites as well. This study that they performed was to interrogate pancreatic cancer cell lines with a metastatic ability. So for this experiment, they had metastases as well as primary tumor samples, and each of which have very good barcode recovery as well as cell recovery, ranging from a few thousand to over 10,000 cells for these sites. And they've also been able to recovered many kinds of clones in one single mouse. So first here, what I did is, first of all, we reconstruct what the phylogenetic trees should be from the single cell CRISPR edited barcode information. And as you can see, there are around 30 clones that are being reconstructed for this single mouse. And there is a variation across these tree structures due to number of reasons. This could be due to the sizes of the clones that's being collected, uh, also barcode recovery rates, barcode editing rates. But on top of that, there is also a clear reason underlying that is related to the evolutionary dynamics. So for the more complex barcoded trees, they are most likely to have come from the more proliferative clones. And in fact, if you look into the data, these clones corresponds to the one with the most mesenchymal properties, which are the ones that are more likely to metastasize. On the right-hand side here is a UMAP embedding of the single-cell RNA sequencing data coming from the primary tumor of the mouse of mouse 1. For this clone, it is the largest clone of all with more than 9,000 cells and over 3,000 leaves. And to apply because of all the variation that I was explaining to you in the barcoding structures, what I want to do is to focus on only the lowest level of these trees, so only the tips of the clonal trees, and examine how many cells share the same unique barcode in the experiment. And what I call them is, I'll call them siblings from now on. And within each of these sibling clades, there is a variation in the number of cells that are assigned to such a leaf. Like you can see here, the sibling count coming from clone 1 can vary from down to 50 to all the way to 250. Although it is possible, and it is definitely a reason that this is due to different barcode recovery. A quick glance at the ranks of how big these leaves are and how it's associated with number of barcodes lost shows that this is not the sole reason, and the largest leaves is not necessarily the ones with the highest lossiness in the barcodes, and therefore we would expect that there contains some information about the evolutionary dynamics and the transition dynamics that has happened in the pancreatic cancer evolution of the mouse. To visualize what these sibling clades look like on this UMAP embedding, here are 3 sibling clades that are the larger ones that I have picked to show you. So you can see that for each of the sibling clades, we have quite a lot of cells there because that's what I have chosen to show you here. But another important thing to note is that you can see that the cells do distribute quite widely over the UMAP embedding. And what that tells us is that the transition that occurs over the evolutionary timescale of the CRISPR edited barcode is quite um, large. So what that tells us is that it is quite possible for us to understand transition dynamics across the entire UMAP embedding using this method by focusing on the lowest level of the tree tip. And what I will do is look at each of these sibling clades and focus on pairwise cell state comparison to start off with. And what that means is that I will look at each of the sibling clade and then count the number of cell states A versus cell state B to show that this is a feasible method and also a informative metric. What I do is now I go back to the evolutionary modeling that I started off telling you about with. So what I have is a birth-death process that describes the evolutionary process of the tumor, meaning that the birth-death process has a Poisson distributed number of offspring for every stem cell over every generation. And the average number of offspring is chosen such that the tumor grows exponentially, just like what you would expect for a tumor. In order to mimic CRISPR editing, there is new lineages being mutated from the population with a unique label under a fixed mutation rate. There is a cell state assignment happening whenever such a mutation occurs, and this is where the explicit modeling of phenotypic transition comes in. For every cell, there is a associated cell state factor, which has a dimension that is equivalent to the number of states. For now, it is n equals to 2, and this cell state factor starts off being 1, 0, or 0, 1. Over time, though, this cell state factor will evolve based on a Markov model that is determined by two parameters, TAB, which is the transition rate of the cell going from cell state A to cell state B, and TBA, which is the reverse transition rate going from cell state B to cell state A. So I have this computational simulation going. And what that means is that towards the end of a simulation, I can have both the genetic phylogeny as well as the cell state of every single cell being recorded. More than that, I could also look back into the history and see how the clone size of each lineage has fluctuated across time, and also how the cell state would have changed for each of these cells across the generations. And like you see here is there is transition going on from cell state A to cell state B. Using this method, I can generate the exact kind of data that I am looking at from the mouse data that I was showing you. So I can look at the genetic relationship across all the cells that I sample from my simulation. I can build a phylogenetic tree and then again look at the lowest level of these trees and count and interrogate the sibling clades. Like you can see here, even though two of the cells share a relatively recent common ancestor, they still have different cell states, and that's because of the phenotypic transitions that have occurred that results in the observation of intermixing on the right-hand side, as you can see. So if in a if in a sibling clade there is two different cell states both being measured, then what I call is there is intermixing, and such a clade is then called inhomogeneous. So this is an inhomogeneous cell state. And if we go in to count every single sibling clade that is simulated and use a simple consider the simple ratio of number of cell states B versus number of cell state A in each sibling clade, we get a number for each of these sibling clade, which can then be converted to a estimated transition rate based on a very simple algebraic manipulation. And as you can see, if I increase the TAB in my simulations down the column, then the simulated data will cluster around the true value of TAB as the true value increases. on the right-hand side, you can see that there is a simple correspondence between the inference of the cell state transition rates versus the true TAB under this multi-state evolutionary model. For a more complex scenario, now if we allow transitions to be reversible, unlike the previous slide, now we have TBA not being a zero number, then what we see is that this metric that I have been describing to you about is a metric that now shows not just what the TAB value could be, but also the TBA value. As we move towards the right, the transitions become increasingly reversible, and we have a TBA that is a TBB that is decreasing. TBA increases, TBB decreases because they sum to one. In such cases, what we see is then there is a mixture of two peaks in this metric measurement. And so what we can, even though this is not exactly what happens in the real world as you would expect, what this tells us is that this metric is an informative metric which contains information about the transition dynamics occurring over the timescale of the CRISPR edits. Yeah, so that's just some more benchmarking showing that the observed inference from the simulation corresponds well with the true transition rates. So back to the lineage tracing data now. We have, so again, we have 30 clones, but for this part of the presentation, I will first start off with focusing on the primary tumor. So looking at the primary tumor, we have a simple, we do a simple clustering, and with the lowest resolution available, we have just 3 to 4 clusters. So it's very easy. We could just start off by assigning one of the cells, one of the clusters being cell state A and the other cluster being cell state B in the primary tumor. And here I perform exactly the same measurement that I have been describing to you. So I first note the number of inhomogeneous clades there are from the lineage tracing data. Then I count the number of B cells versus the number of A cells. And on top of that, I also look at the number of homogeneous clades. So in total, there are 67 inhomogeneous clades, whereas the remainder are homogeneous clades, summing together to 199 total clades. And so by doing again a conversion from this metric, there is clearly transition flux occurring between A and B. And that's actually very intuitive, because that's just telling you that there are cells with explicitly different cell states that share a very recent common ancestor. So therefore, there should be transition in the phenotype space that has occurred. Just some more demonstration of a different way of annotating the cell states. We could also do the cell state assignment by using different kinds of stem signatures, epithelial, mesenchymal, gene signatures, and using different kinds of looking at all the permutations in this slightly higher resolution clustering. You could see that the green and the orange shares more transition flux than the green and the blue. Similarly, for the orange and the blue, they share more transition flux than green and the blue, which is as expected from what we know about the RNA velocity. So using RNA velocity methods, we have checked that the latent time goes from the green cluster to the orange cluster to the blue cluster. So it is within expectation and also consistent that we are seeing less transition flux between the green and the blue that have been realized over the time scale of the CRISPR edits. Another thing that provides us with information is the number of homogeneous clades. So for example, here we have 67 homogeneous clades coming from the green cluster, but none coming from the orange cluster. And what that could be telling us is that there is the potential ancestral state of the sibling clades that we have harvested are all coming from a ancestral state A. So before we do all of this more systematically, there are a few other effects that we need to consider. First of all, is that in our metric, there is discrete effects coming from non finite sizes of the leaf at each of the, so because each of the leaves have a finite number of cells, right? So it is possible that this discrete effect can affect what we see in as our null expectation. And other than that, we also have sampling effects coming from whether or not we would observe exactly the expected average based on the bulk information, the bulk ratio of B cells versus A cells that are being collected. So after considering these effects and doing this more systematically, we want to quantify whether or not there is clear signal telling us that the null should be rejected and there is a transition flux occurring across different clusters, different cell states. And so can we infer transition flux across arbitrary cell states? Can we map the clusters, look at the clusters on the left-hand side, combine it with the lineage tracing data, with our multi-state evolutionary model, with our numerical methods, can we then infer a transition flux between the different states? And so building on what I've been telling you, we now have a null expectation that's calculated for both the number of homogeneous clades as well as the number of inhomogeneous clades. Our null expectation is that for every leaf, so for every tip of the tree, we have a binomial sampling process that occurs with an expected average that is based on the total ratio of B cells versus A cell across the bulk sample. And for the inhomogeneous clades, what we do is now for sibling clades that has a clear intermixing of the two different cell states, what we now do is we again look at the ratio of number of B versus number of A. So that is the orange curve that you see on the top right plot there. And we compare this curve with what is expected if we instead had a binomial distribution in combination with the consideration of our leaf size distribution, what would be the difference in the distance across the two? So that's just a metric telling us how different these distributions are. And this metric is used as a primitive metric for indicating the signal that is present across the two cell states. We then weight it with the number of clades supporting this signal and use it as a method of quantifying the transition flux between the two cell states. The number of homogeneous clades is also important, and what we use it is for postulating the direction. So if we have a number of observed A clades being way more than the binomial expectation, homogeneous B clades that's observed being way less than the binomial expectation for B clades, then we would postulate that the ancestral state was A, and therefore the transition flux comes from A to B. So doing that, and here are just a few examples across several arbitrary pairs of cell states, we could see that there is a strong indication of the blue cluster being the ancestral cluster with flux transitioning into other neighboring cell states there. And this is consistent with RNA velocity analysis, where the upper cluster corresponds to the earlier latent times for the cells. Comparing this analysis with, again, the gene expression across these UMAP embedding, you notice that there is a clear upregulation of stemness signature in cluster 3 and cluster 4, so the red and the purple clusters on the right-hand side. And what we are observing is that there is a strong shared genetic relationship between cells coming from this cluster with cells coming from cluster that is colored blue here, and cluster that is colored orange here. And what this is telling us is that there is a clear transition flux across the boundary going into or out of stemness. While we need to do more vigorous analysis to verify that there is indeed potential signal of dedifferentiation in cancer cells, this is already a very good support for the observation that it is a frequent case that in cancer evolution we would observe the transition flux occurring across the stemness boundary. Yes, so for other metastatic sites, we've also performed the analysis, including the metastases that are found in the liver site and the peritoneal sites. At the surgical site, we have a very similar UMAP embedding of the cells that are being harvested. However, the signal is not so strong for us to recover any sort of differential gene expression for stemness. So we weren't able to find the same transition flux across the stemness boundary by looking at the surgical site sample. And this could be due to the different recovery of different clones and different number of cells being harvested. For peritoneal metastases, if we look at the gene expression, we see that there's a very interesting differential gene expression profile for the epithelial markers and the mesenchymal markers, with the orange cluster being more epithelial and the green cluster being more mesenchymal. And combining that with our lineage tracing analysis using my method of inferring transition flux, we can see that the two clusters are related by sharing a common ancestor coming from the blue cluster. And then for peritoneal metastases, the gene expression profile is maybe not as interesting for our lab's purpose of understanding stemness evolution, but we could also see that there is some clear transition coming from the different clusters. Yeah, so if we look at all sites together, because we had separate sampling processes, my method needs to be adjusted. So for now, what I'm just showing you here is a non-directional transition flux. And again, we could see that for the peritoneal metastases and the liver, there is a lot of sharing of recent evolution with the primary tumor. So, so thanks for listening about my ongoing work right now. What I have shown so far is that intermixing of cell states at the sibling clade level is a metric that allows us to interrogate the phenotypic transition rates using the lineage tracing data that is becoming increasingly available. And this lineage tracing data is also something that our lab is trying to generate at the moment, focusing on liver cancer instead of pancreatic cancer. Yeah, and then there is so far, we are able to see some transition flux across phenotypic boundaries that are relevant for stemness and dedifferentiation, which is an important phenomenon that nowadays we are trying to understand better in order to know the prognosis for a patient. And for future work, more systematic um, analysis will be done. We will use Gaussian mixtures to pick up subpopulations within the samples that we've collected, corresponding to probably different behaviors of the underlying evolutionary dynamics. And other than that, also we will use Bayesian inference combined with the multi-state evolutionary model for better quantification of the exact transition rates. And thank you for listening. That's the lab that I'm working with right now. My PI is Professor Stephanie Ma, and if you're interested in collaboration, please drop us an email.